All right, so now we're going to switch to what's my favorite topic in biochemistry, which is amino acids and proteins. Um, First, we're going to just start by introducing them and then introducing the various like kind of classifications of proteins. Start by just introducing amino acids here. First, just like a little bit of background, sort of whatever, skip that. Basically, what we're building up to is this idea of proteins. Proteins are all made up of the same 20 building blocks, which are the amino acids. So again, the amino acids are going to be the focus of this particular lecture, but just some sort of background about why we care. Proteins are like essentially how I think about them is they're like the worker bees of the cells. They really kind of do almost everything. I mean, we talked about how carbohydrates are important for energy and lipids are important for storage of energy as well as making up membranes. Um, the actual like activity of the cell is done by proteins. So when we talk about sugars being metabolized, that's a protein breaking it down. When we talk about DNA being read, that's a protein doing that. DNA being packaged, DNA being made, sugars being imported into the cell, sugars being linked together to form starches. All of that stuff is done by proteins. In addition to that, they also form the major structural components of cells. So we talked about cellulose as being a specific structural component that's made from sugars, kind of unique to plants. Most structural components are proteins. So cartilages, muscles, hair fibers, your fingernails are all proteins. They're just really central to the metabolic functions of a cell. And again, one of the crazy things is that they are made from the same 20 amino acids from any organism, from something as small as a virus or a bacteria to a fungus to a plant to a reptile to a bird to a human. All of them have the exact same 20 amino acids that they're working with. Um, they're just linked together in different ways, and that forms all these different tools at a cell's disposal, which is just pretty wild. So if we're going to sort of classify proteins and their functions, so again, structurally, collagen is a big one. That's sort of in your more flexible cells compared to hair and skin, which is keratin. Your contractual proteins, so which makes muscles move. Transport proteins, so the things that are responsible for getting the sugar into the cell or getting a waste product out of the cell, that would be a protein. We talked about how starches are used to store energy. Your body actually stores energy in proteins as well. Um, and then as well as hormones. So insulin is a very famous hormone that's actually made up of a protein. We talked about how some hormones or sort of steroids or lipids, that's one class, but there's another one that's also proteins. And then the big ones being the enzymes, which are really like the worker bees of the cell. They're always doing something, they're catalyzing chemical reactions. And then protection, there are proteins that are responsible for recognizing foreign substances. So your immune system is based on proteins as well. All right, so let's take a look at the anatomy of an amino acid. They all share kind of the same basic features, which is if we kind of just pick apart the name here, amino, because they have an amine group, and acid, because they have a carboxylic acid group. Okay. And I'm gonna clarify in a second here, fib on this a little bit, but that's sort of the basis of where the name comes from. In addition, there are two other features. One, those two are separated by this single carbon here, which we call the alpha carbon. Right. So you have the amine group, the alpha carbon, and then the carboxylic acid. And then the other main feature is the R group. And this is what differentiates all those different amino acids is the identity of the R group. Is it a methyl group? Is it an ethyl group? Is it something more complicated like a thiol group or et cetera, right? So that the thing that differentiates these various amino acids is the identity of what that R group off of the alpha carbon is. Okay. Now, I kind of lied in saying like amino acid is a little bit of a misnomer because the amino group is not actually in the amine form, and the carboxylic acid group is not actually in the carboxylic acid form in the cell, right? In what we would call physiological pH, which is the pH that your cells are around 7. Around pH of 7, the carboxylic acid is actually a carboxylate group, right? Meaning that that hydrogen has been lost. So let's actually zoom in here. The hydrogen has been lost, right? So we're not a carboxylic acid, which would look like this. We're a carboxylate. So this hydrogen has been lost, and you have just a negative charge there, right? Remember, the acid form only exists at really low pHs. Otherwise, that acid's been donated and it's a carboxylate. Likewise, the amino form is not actually an amine, it's an ammonium group. So instead of being the NHH amine group, it's now accepted a proton and has a positive charge. Okay, remember, amines can play the role of bases, so they'll only ever be in the amine form at high pHs. Around that physiological pH, it will actually be an ammonium ion. So amino acids are kind of a misnomer. They should be really called ammonium carboxylates or something like that, but people didn't really appreciate this back when it was being named. But it is something that we have to know and to consider. If you're being asked to draw an amino acid at physiological pH, you want to make sure to not draw the carboxylic acid form, but the carboxylate form, and to not draw an amine group, but an ammonium group. Okay. And this particular one, and we'll talk a little bit more about these R groups in a second, but remember, these R groups are what differentiate the amino acids. This is just a methyl group. This is one of the simpler amino acids. Okay, but so anyways, this is something that's really, really important to keep in mind, all right? The fact that you actually have an ammonium carboxylate, not really an amino acid. So that name's kind of a misnomer. The reason that's important for you guys is that this is the constants page that you're going to be given for your exams. This is a really typical way of looking and listing amino acids, is something like this. And if we zoom in real quick, this is that same one. You see it's not in the correct form. Okay, so this is what you're going to have on your exam, and you're going to want to know that actually this hydrogen shouldn't be here, should be replaced with a negative charge, and that this isn't NH2, but rather we should replace that as NH3 plus. Okay, so that's an important thing that you guys are going to have to make sure that you know. For not only my exams, but the ACS exams, is knowing, okay, given this information here, I know that it's actually an ammonium carboxylate, not an amino acid. It's just an unfortunate misnomer that has survived throughout history. Okay, and we'll talk about these different amino acids here in a second. But again, this is the constants page that you can expect, which is just an alphabetical listing of all the amino acids. One of the things that I'll point out, and actually zoom back in here, is for each amino acid, you're given 4 pieces of information. Okay, I'm not expecting you guys to have these memorized for the exam. You might have to do that in a future course, but not in my course. I'm not worried about that. You just need to know how to use this. Okay, 4 pieces of information. The first being the structure. Okay, the second being the name. So this would be alanine. And then each amino acid has 2 different abbreviations that are used. The 3-letter abbreviation, which for alanine would be ALA, and the single letter abbreviation A. Okay. For some of these, it's pretty straightforward. ALA, alanine, A. I mean, like, you could see how those are all related. For other ones, if we just move over one, this is now asparagine. You see that it's 3-letter abbreviation is not ASP like you might expect, it's ASN, and the 1-letter abbreviation is an N. Who knows where that comes from? Okay. So some of them are pretty obvious. They make sense. Other ones are a little bit more complicated. Again, I'm not expecting you to memorize these, but it is something to be aware of. It's not just the first three letters and then the first letter all the time. Okay. Um, but yeah, so, and we're going to go into these different R groups here in a second. But again, the charge state of the amine group as an ammonium and the carboxylic acid group as a carboxylate is really, really important. And again, the alpha carbon is what that R group is bound to. All right. So we can categorize as our amino acids based on their R groups and the properties of their R groups. Some R groups behave very similar. Some of them are very different. Okay. There are non-polar R groups. Those are maybe as small as just a hydrogen or an alkyl group we know is a non-polar group. Also an aromatic, something like a benzene ring would also be a non-polar group. In biochemistry, especially protein terms, a word that's synonymous with non-polar is hydrophobic. Hydrophobic is just anonymous to non-polar. And if you pick it apart, it means water fearing, right? So these are groups that don't like water, okay? Because so much chemistry of proteins takes place in the cell, which is largely water, we kind of, instead of referring to these groups as non-polar, often refer to them as hydrophobic groups. And then there are a few different types of polar groups, okay? Polar groups would then be what are called hydrophilic, right? Hydrophilic is then synonymous with polar, just like hydrophobic is synonymous with non-polar. Polar groups like water, hydrophilic, water loving, right? There are three categories of polar groups. Those that are neutral, meaning they have no net charge. So those would be things like hydroxyl groups or alcohol groups, a thiol group if it has an SH, as well as amides. So if I just want to draw this out, remember an amide, something like this, would also be a polar group, but it has no net charge. There are polar acidic groups, and these are carboxylate groups. So again, a little bit of a misnomer in that they're not the acid form. They're after it's already donated that proton. So polar acidic groups have a negative charge, and they actually all have the same carboxylate group on them. Okay, so we call it acidic because it's a carboxylate group, but it's after that sort of donation of that proton has occurred. And then lastly, we have these polar basic groups. And those all have some form of an ammonium ion or a protonated nitrogen group there. So again, we call them basic, but they don't actually play the roles of bases. It's already after they played the role of a base. So they've already accepted that proton. Well, they look a little, the three of them look a little different from one another than the acidic groups. The acidic groups all have that carboxylate group. They all have protonated nitrogens, but sometimes that nitrogen has a double bond or is in a ring or something like that. So they do look a little bit different than the polar acidic groups, but nonetheless, all of those would be considered polar groups. Remember, a net charge, like a net negative or a net positive charge, is really, really polar, as well as anything that can form hydrogen bonds. So that's why the thiols and the alcohols and the amides. All right, and we talked about the abbreviations. All right, so let's look at a few of these. We're just going to go through each one of them. Again, you don't have to have them memorized, but it's a good idea to just kind of lay eyes on them so they don't look too terribly foreign. These are actually grouped based on those properties that we were just talking about, the non-polar nature, et cetera, et cetera. One of the things that I want you guys to do is to go through this page, this constants page, which is what you're going to be given for an exam, and to make sure that you can categorize them based on their functional group, because this is what you're going to be given for an exam. That's a really important skill to have. This is in alphabetical order, though. It's not going to be grouped based on the properties. So you want to make sure that you can look at those functional groups and tell which category they're supposed to go into. Okay, so let's start. We're going to go kind of the simplest ones here, which is the nonpolar R groups, which have either just the hydrogen, the alkyl groups, or an aromatic. All right, so glycine is the simplest of the amino acids in that it doesn't really have an R group. It's just a hydrogen. It's really teeny tiny as far as R groups go, right? Because it's just that hydrogen there. It's considered to be nonpolar because, of course, that group doesn't, just a hydrogen can't form hydrogen bonds. It has to be like an OH or an NH. The next simplest one is alanine, which has a methyl group. And then there are ones of increasingly larger alkyl groups. So you have valine, which has, kind of this fork going on. It's a 3-carbon alkyl chain. Leucine, which has a fork, but it's a 4-carbon chain. And then isoleucine, which is again 4 carbons, but the branch comes at the first carbon instead of the second carbon. All of these are just alkyl groups, right? There's no fancy functional group going on. So these would all be considered nonpolar. And let me just point out that your book is actually much better than like my textbook was when I was in school. Really good at making sure to picture these amino acids as actually ammonium carboxylates, right? So with a charged nitrogen and a positively charged nitrogen and a negatively charged oxygen, right? So this charge here and this charge here are really important to keep in mind. I kind of stressed that earlier. Other nonpolar groups would be aromatic groups. So something like just a benzene ring on phenylalanine. This is a big nonpolar. So now we're getting kind of a little bit larger with these aromatic groups. Methionine is a little bit of a weird one in that it's kind of like an alkyl chain, but there's this sulfur here. It's still a nonpolar group. It can't really form hydrogen bonds. So it's not a polar group. Proline is probably the weirdest of all the amino acids in that it looks a little bit differently. And I don't think that this drawing right here does it justice. So I want to show it instead on our constants page. Go to P for proline. You see that the from the alpha carbon, and then if we go all the way around to the amino, which is really an ammonium group, it's all locked in this ring. Okay, so both the alpha carbon and that nitrogen are part of this 5-membered ring. Prolines are considered nonpolar groups. They also tend to be found at very rigid points of the molecule. Unlike some of these other amino acids that can sort of rotate freely, proline is kind of locked in a particular orientation because of that ring. So it's usually present at certain turns and something like that within the protein structure. It's the only one that's like that, but it is a little weird and something to keep in mind. And again, I just, it's well illustrated here, but I just want to point out from our constants page, you want to keep in mind that this is not correctly drawn as is. Really, we should get rid of this hydrogen here and make it a negative charge on the oxygen. Likewise, we should put a second hydrogen on this nitrogen and have it be a positive charge. Okay, so that would be the correct structure for proline at physiological pH. And the last one is tryptophan, which is a huge functional group, or huge R group. It's not only a benzene ring, but then also attached to a 5-member heterocyclic ring. It does have this group here, which could be capable of forming hydrogen bonds, but the butt end of that huge benzene 6-membered ring and the 5-membered ring, there's always that push-pull of a polar group and a non-polar group. The non-polar part is just too big to sort of overcome the properties of that polar group. And so this is considered to be a non-polar group. All right, then going to our polar groups that are neutral. So serine and threonine, they both have hydroxyl groups or alcohol groups. Um, Tyrosine has like a phenol going on, so it does have a benzene ring, but then it's attached to that hydroxyl group, and we categorize this as a polar group. And then cysteine has a thiol group, which is similar to an alcohol, not quite as good at forming hydrogen bonds, but still pretty good. And then we also know that our amides are good polar groups. And so there are two amino acids that have amide R groups, and that's asparagine and glutamine. So again, both of these have amide functional groups. There are two acidic polar groups. And remember, this means that they have a negative charge associated with them. Both of them have this carboxylate group, and just on different lengths of a carbon chain. So aspartate has a 2-carbon chain, and glutamate has a 3-carbon chain, with that last carbon being a carboxylate. And importantly, so let me actually erase here real quick. This is again where you're going to have to keep in mind what the structure is at physiological pH. Just like this carboxylic acid group has a negative charge, so does our R group that also has a negative charge. And then of course the amine is actually an ammonium and has a positive charge. Something to keep in mind, because if we were to look up glutamate on our constants page, it's not glutamate, it's glutamic acid. So it's just it's the same molecule, but not given to you as the form that it is at physiological pH. So we have to know that this hydrogen, just like we said before, is gone, and that this is actually a negative charge. Likewise, it's not glutamic acid because this hydrogen's gone, and it's that negative charge. And that this shouldn't be a 2, but a 3, and that there is a positive charge. I'm going to be honest, your textbook is pretty good about giving it to you in this physiological state. However, I know from experience that's not how it's usually given. And so although they're telling you the amino acid is glutamate, truthfully, you will see it referred to as glutamic acid in the literature way more. These are historical kind of pain in the butt reasons why that's the case. Again, the whole like charged state thing wasn't at physiological pH wasn't really appreciated at first. That's why I want you guys to get in the habit of being able to look at this group here in its carboxylic acid form and saying, no, I know from what I've learned that that's not a carboxylic acid, that is a carboxylate. Okay, so that is one of the skills that I'm trying to train you guys to have that I think is really important for the ACS exam. And then there are 3 polar basic groups, and these all have protonated nitrogens. Again, they don't all look the same, so let's look at these. So first of all, histidine has this, it's this heterocyclic amine ring where one of the nitrogens that's in a double bond has an additional hydrogen on it. And so that has a, let me get a blue going on here. So that has a positive charge. Lysine is probably the most straightforward one because it's just a normal old amine group that's been protonated. And so that has a positive charge. And then arginine actually has 3 different nitrogens going on. And one of them, the one that's double bonded, has an additional hydrogen and a positive charge. So again, let's find these on our constants page so we can sort of see what they would look like in the material that's given, because again, they're not given in their charged form. So histidine, if I was to draw histidine at physiological pH, I would, just like always, I would make sure that this oxygen actually is not a carboxylic acid, but a carboxylate, that this amine group is actually an ammonium ion. And then lastly, that this nitrogen has a hydrogen attached and a positive charge. I don't know, maybe, hopefully I'm not going to confuse anybody here. I will say there's another nitrogen in this ring, right? So how do you know that it's not that the extra hydrogen isn't there? Truth be told, it's always over here, but that's not a huge mistake to make. Both of them could possibly accept that hydrogen. It just turns out that that one that has that double bond is more likely to accept it. All right. Important, I know that I've been going over and over this with the making sure you add the charges to the nitrogen. One thing that you should keep in mind that I think is an easy mistake to make is what type of functional group is this? This is an amide. Amides do not have positive charges on their nitrogen. This would be incorrect to draw it like this, all right? Because amides can't play the role of bases, only amines. So remember, we talked about these amides as being neutral polar groups. So don't make the mistake. This is dead wrong to put that positive charge like that. All right, so for this, if I wanted to make this one at physiological pH, the only thing I would have to change would be my amino group to an ammonium and my carboxylic acid to a carboxylate. Don't have to touch the R group at all. Arginine was another one of these basic functional groups, or they have a positive charge. And so again, this is what you would see for arginine. Like always, we're going to get rid of this hydrogen here, put a negative charge. We're going to make this amine group into an ammonium. And then for arginine, it's the double bonded nitrogen that gets that positive charge. So this would be the correct form. This is how I would have to modify what's given to me on my constants page to give you what arginine looks like at a physiological pH. And then the last one was lysine. Here we go. And lysine is just a good old amine group as the R group. And so just like we changed this to an NH3 plus, we would do the same thing to our R group. And then remember to get rid of this hydrogen and put a negative charge. So this is how you would draw lysine at physiological pH. All right, so now let's do some more here. So again, this is the type of question that you might expect to see. Draw serine and aspartate at physiological pH. Okay, so serine is what the first one I'm going to find. Oh, I guess they're in alphabetical order. All right, so here's serine. We have our alpha carbon attached to one other carbon and then a hydroxyl group. I'm going to remember to put my carboxylate and my ammonium. And then we had 1, 2. Right, the 3-letter and 1-letter abbreviation, again, that's not something I expect you to know. So all you got to do is be able to look it up correctly on the constants page. So this is serine, which is ser and S. Now, aspartate, so this is another one. Remember when we saw glutamate, it was actually glutamic acid on our constants page. We're going to have the same thing here. Aspartate is actually aspartic acid on our constants page. So these are the two acidic functional, acidic R groups that you need to know. Your book does a good job of referring to it in the correct physiological pH form. That's just not going to be the case, like normally, honestly. aspartate is the deprotonated form, but the name of the amino acid is aspartic acid. Glutamate is glutamic acid. Okay, so that's just something that we want to be good at, basically, is knowing what's being talked about there. So anyway, aspartic acid is 2-carbon carboxylic acid functional group. That's pretty bad. Again, I'm going to draw my it's not an amino acid, it's ammonium carboxylate. So there's a carboxylate group there and an amino group there. And then this is a 2-carbon carboxylic acid group. And I'm going to remember that at physiological pH, those oxygens, those oxygens have that negative charge, just like it did in this carboxylic acid group is actually carboxylate. Same thing up there. Okay. I don't want to get too much into like this particular health defect other than to say there's one property of the cysteine amino acid that I think is important to note. It's something that we're going to see later on as well. It turns out that if you're a disease in which your body produces too much cysteine, let's look at cysteine again. That's this one with this thiol group. If your body produces too much cysteine, you get these kidney stone-esque things, except for they're not formed in, they're not the same material as a normal kidney stone, and they're actually quite a bit sharper, and it looks pretty awful. And they form because cysteine has this property where 2 cysteine amino acids together can be reduced to form this type of linkage here, right? So going from this, we have 2 thiol groups into what's called a disulfide bond, where those 2 sulfur groups, a different color. 2 sulfur groups lose their hydrogen and form a single bond between them. Yeah, so this is pretty terrible if it happens in the wrong place, like in your bladder or in your kidneys. Although this disulfide bond is also something that holds together proteins in a legitimate way. We're going to see it's a pretty essential part of some protein structures. So this is a disulfide bond that can take place between 2 cysteine amino acids.